Hello. Mm, glad to see you. It is I, your old nemesis, Wolbesha. I will not be your guest speaker today. I am just stopping by to say hello to my friend E. coli. Hello. Hello, old buddy. You old so and so. Well, we're not really that good of friends. Uh, I also wanted to stop by to tell you, do not wash your hands. Be sure to stand right next to all sorts of strangers, especially those who are coughing, and don't get much sleep. No, just stay up all night. Okay, kiddos, see you later. Well, thank you. Thank you, Wolbesha. That was very informative. Let's begin. Today we're talking about antimicrobials, which I have a hard time saying. Let's see. Okay. Okay. Um, Antimicrobials, Chapter 10 in Veterinary Pharmacology. I hope you're all doing well. I imagine you guys are under a lot of stress. So please let me know what I can do to help. Hang in there. We're going to get through this. If you're familiar with the TV show Survivor, you know it's about a bunch of individuals in a kind of hostile environment and they have to outwit and outplay and outlast each other. Well, antimicrobials are kind of the same thing. Although in this case, it's actually antibiotics versus bacteria. And we do not want the bacteria to outwit and outlast us. We want the antibiotics to outwit and outlast the bacteria. And that's part of what we're going to talk about today. We're going to learn a lot more about bacteria and about these weapons we have. But there's probably a lot that you already know about bacteria and possibly antibiotics. You may have heard of things like strep throat. That's a bacteria caused by streptococcus. You may have heard of staph infections. Staph with a PH stands for staphylococcus. There's several different kinds of staphylococcus. A urinary tract infection, that's bacteria. I had acne as a teenager, and that's caused by a bacteria, and I had to take antibiotics for it. We're also, not this week, but next week, we'll talk about another microbial known as the different fungi, or however you say the plural of fungus. But this week, we'll be talking about bacteria. And we'll also be trying to explain the differences between some bacteria. The ones you just saw, strep and staph, they're bad. Some are even worse. There are ones that have been in the news, like E. coli, killed a number of people back in the early 1990s. Salmonella shows up in some other food recalls. So, and these can cause, these are food related problems, but we see E. coli causing bladder infections. We see salmonella causing some really horrible gastroenteritis problems. So we'll talk about how these different bacteria are a little different and how you can learn to predict one type or another. If someone says gram negative, you should feel a cold shiver down your back, and we'll explain that a little bit more. Let me just look at my notes here real quick. Okay. Yeah. So our goals today are going to be, we want to describe some of the terms that you'll hear when discussing these drugs. Terms like cidal, like bactericidal versus bacteriostatic. There's these other terms, concentration dependent and time dependent. We'll talk a little bit about those. We're going to want to learn how to identify appropriate antibiotics based on culture and sensitivity results, describing the beta-lactam ring of penicillins. That'll help. And the role of clavulanic acid. We'll describe some differences between gram-positive and gram-negative bacteria. That'll help you make sense of the different antibiotics. And when we discuss part of this, especially culture and sensitivity and these other terms like minimum inhibitory concentration and resistance, we're going to tie it in with a real story about a cat named Gordo. It's going to feel a little bit like a tangent, but I'm trying to explain it a little better than chapter 10. Chapter 10 is not bad, but it's parts of it are a little dense. And so I'm just trying to show you a different way of explaining it. Resistance, that's the topic in your discussion this week. So we're going to talk in detail about how resistance develops and emerges, why it's a concern, and what can be done about it. And you'll be talking this week about partly about our role as members of the veterinary profession. And then we'll talk about the basic mechanism of action and key characteristics and disadvantages or adverse reactions to three families: the penicillins, the cephalosporins, and the aminoglycosides. So again, just to kind of begin, there are many different kinds of bacteria. They show up in different places. They cause different problems. Some are worse than others. Some are little balls, kind of like this, right? So we get some that we call cocci. If you can see that. Things like streptococcus, staphylococcus, they all have that word coccus, which is a ball. Other kinds of bacteria we'll call rods. They're also called bacillus. They're kind of long, like this balloon here. This streptococcus, that's not really a great example. When you think of rods, it's better to think of things like salmonella or E. coli. Some of these are human diseases, but basically we've got either rods, kind of long rice-shaped bacteria, or we get these little balls. And then there's some that are rods, things like the agent that causes tetanus. Here's the agent that causes botulism. So, and a term you'll see in the book that you could probably use a review on, or you may never have heard before, is this term gram negative versus gram positive. Now, when we get a sample, maybe a sample of a bacterial ear infection or a skin infection, we can put it on a slide, and then we subject it to a series of stains of different colors. And then we can look under the microscope. And if we see that they are this deep purple, that means they stained purple, and those are called gram positive. And again, the naming has to do with the wall of the cell. If they do not take purple stain, but they take this pink stain, then we call them gram negative. And the reason that's important is it'll help you predict which bacteria are worse than others. Gram-positive bacteria are usually more predictable and more easily killed than the gram-negative bacteria. So Streptococcus, Staphylococcus, they can be bad, but they're usually kind of predictable. The same antibiotic will usually work. Um, um, there is one exception, and that would be a kind of Staphylococcus called Staphylococcus aureus, specifically one called methicillin-resistant Staphylococcus aureus. That one is not so easy to kill, and it's not so predictable. And you may have heard it as MRSA. And we'll talk more about MRSA today. Gram-negative bacteria tend to be very unpredictable. It can be the antibiotics you think might work, they might not work. So Salmonella and E. coli are two. They were, I believe, one or the other was involved with Chipotle, had some food poisoning issues last couple of years. Pseudomonas, you learned about when we studied disinfectants. Pseudomonas can make that biofilm, that kind of slime that protects it from different antiseptics. That's a really nasty bacteria. It's really hard to get rid of. Anyway, just keep these descriptions in mind as we go forward. And you'll see that some antibiotics are more effective against gram-positive bacteria, and some are more effective against gram-negative bacteria. So if a patient had an infection with E. coli, we would want something that was effective against gram-negatives. And you'll see in the lecture, you'll see I'll use G+ to stand for gram-positive, and G- will stand for gram-negative. Well, bacteria, the other thing that's important to know is these guys divide quickly. Maybe not as fast as this, but these little stinkers can make more and more and more photocopies of themselves within a very short period of time. If you have not watched the videos in the Start Here page, then stop and watch those, because they talk about resistance. And you can see if bacteria divide pretty fast, it's pretty easy for them to make, they have a lot of opportunities to swap plasmids and genetic material and share resistant genes and things like that. And again, let me look at my notes here. All right. The part of our behavior and part of the importance of studying antimicrobials is the knowledge that some of them are not working as well as they used to. You'll hear about superbugs and antibiotic resistance. I like the point it makes. It's not coming, it's already here. I have an antibiotic that I was using, can't really see it. This is just plain old penicillin, the first antibiotic ever discovered, just about 35 years before I was born, so back in 1930 or so. And they hardly use that drug in humans anymore. Luckily, we can still use it in animals, but penicillin doesn't even work on most human bugs, most human bacteria, because of resistance. So anyway, these are just some of the important things we want to keep in mind when we're talking about them. And our role, lots of different factors play a role in resistance. Certainly when we treat livestock, well, those antibiotics end up in the urine and in the poop, and then that can end up being spread around to people, dogs and cats and chinchillas and snakes. It ends up in their urine or waste products. We use antibiotics in fish, and all of these things that can end up back in you, and without you knowing it, it's killing off the more wimpy bacteria and leaving behind bacteria that have outwitted and are outlasting our antibiotics. And I said earlier, penicillin was only invented about, I'm 52, and it was invented in 1929. So that's only about 35 years before I was born. Before that, there were no antibiotics. A paper cut could kill you. There was a famous athlete in the 1920s. He got a blister on his foot. It became infected, and he died. That was what life was like. Sexually transmitted diseases that now you would just go to a clinic and get some antibiotics, back then could kill you or horribly disfigure you. Women and children routinely died in childcare. In childbirth. Now, of course, that almost never happens. For tuberculosis, the treatment was fresh air, right? Good luck if you have tuberculosis. That's as good as it gets. So the concern is that we may end up back in a world where antibiotics don't work. We have to, hopefully we can stay one step ahead of the bacteria, but we need to be mindful of our practices. Okay, so do any of these look familiar, right? These are some of the antibiotics we'll talk about today. When and if you can return, not if, but when you can return to internships, or if you are working at places, look on the shelf, see if you recognize any of these, things like amoxicillin. Here's the same antibiotic, here's the same one that I just showed you right here, that agricillin. It says penicillin G procaine. There's something called procaine added to it. Ampicillin, those are all penicillins. Over here we've got, oh, and down here's another penicillin. It's amoxicillin that's had clavulanic acid added to it. I love clavulanic acid. Over here, this one called cefovecin and cephalexin. Those are members of the cephalosporin family. We'll talk about them. And then this one has, Neosporin has something called neomycin in it. And that's a member of the aminoglycosides. And we'll talk about that, of course. And I just threw that in there because augmentin, you might know as it's used in human medicine. The same drug in veterinary medicine is called Clavamox. So just so you know. Okay, so first of all, what are antimicrobials? Well, obviously, these are compounds we're going to use to kill or inhibit microbial growth, either bacteria or a fungus. The book mentions like antiprotozoals, but that may show up next week, but we already discussed that with the antiparasitics. The how we talk about the antimicrobials is partly based on what they do. Do they kill or do they slow down the growth of a bacteria? So we would say some antibiotics like tetracycline is bacteriostatic. It doesn't kill bacteria, it just slows down their growth. Other antibiotics, the ones we're learning today, they're all bactericidal. They're actually going to kill bacteria. And there's actually nothing wrong with being bacteriostatic. The difference, though, is for bacteriostatic, we're relying on a healthy immune system, like shown here, to actually do the killing. We're slowing down the bacteria, and then your or my immune system will kill the bacteria. You have a pretty amazing immune system. But this also means you can't use these on patients with a compromised immune system. a cat with leukemia virus or a cat with feline immunodeficiency virus, you would probably want to use a bactericidal drug. A dog with diabetes or hyperadrenocorticism or cancer, right? Any patient whose immune system might not be tip top, we should probably use bactericidal drugs. And again, the goal is to either kill or inhibit some specific pathogen. How do you choose an antibiotic? You go to a veterinary hospital and there may be, there may just be hundreds of different antibiotics on the shelf. Maybe not hundreds, but certainly 15 or 20. How does the veterinarian select the antibiotic? Well, there's different factors that we look at. So one is, can it get to the site of infection? You remember from week 2 or 3, we learned about how certain parts of the body have barriers. The brain has the blood-brain barrier, those tight junctions between endothelial cells and P glycoprotein pumping things out. The prostate and the eye, for different reasons, are difficult for drugs to penetrate. So you'll see there are certain drugs that if you had a prostate infection, if you had meningitis, these drugs just wouldn't work. Instead of prostate infection, I should say prostatitis. Certain drugs like the penicillins, they cannot reach the brain. They cannot reach the prostate. The flip side is urine. Some drugs are able to achieve really, really high levels in urine, and it makes them great for treating kidney infections and bladder infections and things like that. So that's one factor. Can the drug access the site of infection? Another factor is the host. You'll see there are certain antibiotics that we cannot use in certain species. For example, in the hamster or the guinea pig or the rabbit, there are certain antibiotics that will kill them. So we'll learn more about that. Other factors are the client. If I ask the client to give a medication, if I have the choice of one pill that's once a day or another pill that's 3 times a day, I'm probably going to pick the once a day because it'll be easier for the client to give. Is the pill really bitter, like metronidazole, or is it can it be made into like is it beef flavored or something that animal will like? Is it a liquid? Liquids tend to be easier to give, but some animals, pills are easier with a little peanut butter. And cost is another factor. But the most important factor you already know is, will it kill this particular bacteria? This is E. coli behind me. There's certain drugs that will kill E. coli and certain drugs that will not kill E. coli. And we say that we want the microorganism to be susceptible or sensitive to that particular antibiotic. And in the best case, that question is answered by doing what's called culture and sensitivity. So with culture and sensitivity, let's say that, I don't know, give me an example here. If Mr. Let's grab Mr. Wolbachia. Okay, let's get Mr. Wolbachia back here. If Mr. Wolbachia, here he is, let's say he had a wound on his little head. Well, I could take a swab, pretend this is a swab, and I can swab the wound, and then I can have what's called a blood auger plate. I meant to grab you one. And I can swab a sample on that, and then I can culture that, or I can send the swab to a lab. the lab can then run a series of tests to tell me whether the bacteria is susceptible or not. And again, our goal is we want to be able to achieve concentrations that will kill or inhibit the growth of the bacteria, but not harm our patient. And we may find that it's just not possible to achieve concentrations in a particular tissue. Now, by the way, a lot of the time when we use antibiotics, we don't do culture and sensitivity. The chapter makes it sound like we're always doing cultures and sensitivities. It would be great to do that. It's expensive. It's about $150 to $200. So most of the time, what we do is we make an educated guess, and it's what's called empiric therapy. But it means we are going to, based on the species and based on the kind of infection, we'll choose what the most likely antibiotic is. And we can do a sample in the clinic and do a stain. That's not a culture, but we can at least determine, oh, is this a streptococcus? Is it a staphylococcus? Is it E. coli? There's certain tests we can run. It just doesn't tell us which antibiotic will work. Even if we do a culture, we need to start the patient on some sort of antibiotic. The culture may take 2 or 3 days, so we will often use empiric therapy to start an animal on antibiotics while we await results. If it's a fairly predictable infection like a cat bite abscess, we're probably not going to do a culture. And sometimes the owner just can't afford a culture. So those are all times where we just have to make our best guess. To discuss this concept of culture and sensitivity and these other concepts like sensitive and intermediate sensitivity and resistance and mean inhibitory concentration, I'm going to give you a short version and then a slightly longer version. Okay, so the short version is this was this cat named Gordo, and he had this wound that just would not heal. So we needed to try a different antibiotic. But rather than just try one after another, I did a culture and sent it to the lab, and they gave me a list of antibiotics, and they said, these will work and these will not work. And so then I picked the one that will work, the one that was sensitive, or the bacteria was sensitive or susceptible to it, and we used it, and he got better. And this is similar to the kind of test they did. Most of you did this test last semester in clinical lab skills, right? This is this plate that has Gordo's, a sample from Gordo swabbed all over it, and then these little discs were put on there, and each disc has a different antibiotic. So this disc, you see, it killed a whole bunch of bacteria, and we can use this little measuring device, and it tells us, oh, okay, his bacteria were sensitive or susceptible to that antibiotic. This antibiotic, it killed some, but not as many. So we can say that antibiotic, the bacteria has intermediate sensitivity to, and this antibiotic did nothing. It didn't kill anything. And so we will say that bacteria are resistant to that particular antibiotic. And I would like to say that's all. That's the end. But there are some concepts that we need to make sure we understand. So we're going to go through this again in a little more detail. Feel free to pause it and get up and stretch. Okay, so again, Gordo was a cat. He showed up with this wound. I put him on Clavamox. Well, no, what did I put him on? I forget what I put him on. I put him on something, and he just wasn't getting better. I think we probably gave him a cephalosporin, and he wasn't getting better. And so I thought, well, why was it not healing? The owner was keeping the cone on. She was doing warm compresses. She was doing everything right. He just wasn't getting better. Well, I thought maybe there's something wrong with his immune system. I was using a bactericidal drug, but who knows? Maybe there's something wrong. So maybe he has the leukemia virus or diabetes. No, no, nothing was wrong. And so I thought, well, there must be some other bacteria growing there, some bacteria that's not responding to the antibiotic. So you just have to picture I took a little swab that, and then sent that to a lab. And then the lab puts the sample on this plate. This is called a blood auger plate. And they start by putting it here, and they do this little technique to make individual colonies grow. And so these little colonies of bacteria are growing, and it turned out to be a kind of bacteria called Staphylococcus aureus. All right, that's great. Well, now I know what the bacteria is. So what? What do I really want to know? Right? I want to know what is going to kill this stupid Staphylococcus aureus. Aureus, by the way, means gold. I don't know why it's called gold, but that is what it means, and it's a nasty little bacteria. Okay, so to find out which antibiotic will work, the lab's going to subject his Staph aureus to a different antibiotics at different concentrations, very weak to intermediate, very strong. And then before we find out which antibiotic is going to kill Gordo's bacteria, I want to summarize and paraphrase some of the parts from chapter 10, the parts in susceptibility, resistance, intermediate, and break points. It's not bad, but I just wanted to try to, it's a little dense, so I wanted to try to describe this a little more, so bear with me. I spent a long time on this part of the lesson, and it's still not perfect, but I have to stop working on it. So as we said, this process of identification and drug susceptibility testing is commonly referred to as culture and sensitivity, or culture and susceptibility. And it can be used to determine the susceptibility of a bacterial strain to certain drugs by determining how much drug it takes to inhibit, either slow down or kill a bacteria. So when it's done, the bacteria are exposed to different and increasing concentrations of different antibiotics. They're trying to find the lowest concentration that will inhibit the growth or kill the cultured bacteria. The lowest antibiotic concentration that's needed to kill the bacteria will be called that drug's minimum inhibitory concentration for that bacteria. Again, it's the lowest amount that will kill the bacteria. So for example, over here, each of these test tubes has Gordo's Staphylococcus aureus, and they're at different concentrations of an antibiotic is in each one. So this one has this, it has the most concentrated antibiotic. This has been diluted by half, and this has been diluted by half again. And then they put that on the plate to see if anything grows. That's a little different from those discs. Here they're just putting it on the plate to see if anything grows. So look, there are bacteria growing here. There's nothing growing here. There's nothing growing there. Right? The only difference is the concentration of antibiotic. Right? This has the least, and this one has the most. So what is the minimum inhibitory concentration for this antibiotic for Gordo's Staphylococcus aureus? Is it 8 micrograms per milliliter, 16 micrograms per milliliter, or 32 micrograms per milliliter? Which of those three is the minimum inhibitory concentration? Well, it's not this, because 8 did not inhibit anything, did it? 8 micrograms per milliliter, the bacteria could still grow. This is the minimum inhibitory concentration for this particular antibiotic, 16 micrograms per milliliter. 32 also inhibited growth, but we're going to use this, whichever drug has the lowest minimum inhibitory concentration, roughly speaking, is the drug we want to use. Now you'll see you can't compare one drug to another. They're not all going to use numbers like 16 or 32. But my point here is just to understand what this term MIC means. This is the MIC for this drug. If we can reach 16 micrograms per milliliter concentration in Gordo's wound, this antibiotic will be great. The culture and sensitivity is going to do the same for all sorts of other antibiotics with totally different numbers and totally different results. And then they're going to send that to the clinic. Now, this gets a little confusing. And so I'm going to hopefully make it a little less confusing. The book uses this term minimum inhibitory concentration in two different ways. So one way is what we just discussed. It's what concentration of an antibiotic must be reached in Gordo's wound to kill Staph aureus. Now the problem though is we don't know if that concentration is realistic or not. It might be toxic to Mr. Gordo. And yes, there's Mr. Gordo looking very surprised, saying, "Don't give me anything toxic." So we want to determine this minimum inhibitory concentration. Is it something we can achieve, or is it going to be toxic to Mr. Gordo? So that's where this other use of the term MIC shows up. So far we've only talked about Gordo's particular strain of Staphylococcus aureus. Now the labs have tested thousands of other strain of other bacteria. They've tested thousands of Staph aureus, thousands of other bacteria against the same list of antibiotics at the same different doses. Now, why are they doing this? They're trying to figure out for each antibiotic for each particular bacteria, they're trying to figure out kind of the dose, they're trying to figure out three things. So one is the MIC of a particular antibiotic that'll kill a particular bacteria at the usual dosage. You know, so some drugs, there's a dose, a range. There's kind of a low range, a high range. And so they want to find out, you know, if a particular antibiotic will kill a particular bacteria. They want to know how much it'll take. So if the MIC for Gordo's Staph aureus is equal to or less than this kind of standard MIC, then that means we can say that his Staph aureus is likely to be sensitive or susceptible to that particular antibiotic. And so let's just kind of read this. This is from the book. All right, so after bacteria from an infection are grown and tested, if the drug for Gordo's MIC falls in this range, then the bacterial infection will likely be susceptible. Okay. The labs also do all these tests to determine the MIC of a particular antibiotic to kill a particular bacteria at the highest possible safe dosage. And here, if we find that the MIC for Gordo Staph aureus is closer to that MIC, well, then his Staph aureus is not, it's not sensitive, but it has what's called intermediate sensitivity. And again, this is where a drug at the highest possible safe dose, if it will still kill this bacteria, then we say that it has intermediate sensitivity. And then the MIC of a particular antibiotic that can only kill a bacteria at a dose higher than the maximum tolerated dose. If we find that the MIC for Gordo Staph aureus is equal to or higher than this MIC, then his Staph aureus is resistant to that antibiotic. So again, we're comparing Gordo's Staph aureus and antibiotic tests against these standards. And again, they use this information. You'll see in a moment how we use it to select an antibiotic. So again, if we find that if after a bacteria is grown, that the drugs fall in this range here, we say they have intermediate susceptibility, where we can still use that drug, but we're going to have to use it at the highest possible dose. And then some drugs, again, I'm going to show you this thing again, right? Right. And then some drugs, they just don't work at all. They might, but you'd have to give such a high dose that it would be toxic to the patient, so why bother? Again, I realize this is a little hard to understand. But let me keep going. So here's another way of looking at it. If the minimum inhibitory concentration, right, that's the minimum concentration needed to kill the infection, if the minimum inhibitory concentration for your patient's infection is lower than this break point here, right, the break point between susceptible and intermediate, then we can say that antibiotic is considered sensitive. And that means the bacteria is sensitive to it. Yay. Okay. If the MIC for your patient, for your patient's infection, for that antibiotic, if it's more than this break point, but less than this break point, then we're going to say the antibiotic has, we'll say it's considered intermediate sensitivity. It's really the bacteria that has intermediate sensitivity, but it's going to be sensitive only at the highest possible dose. And finally, if the MIC for your patient's infection is greater than this break point, then that means the antibiotic just is not going to work. The dose would be so high that it would be toxic to your patient, and in fact, it might never work. So that would mean the antibiotic is considered resistant. Although again, it's really the bacteria that are resistant. Okay, so we send that swab to the clinic, to the lab, and we get a report like this. And it's just telling me the MIC for Gordo's strain of Staph aureus for amoxicillin, 16 micrograms per mil will kill it. For amoxicillin, clavulanic acid, it's 4 micrograms per mil. That's the minimum amount that will kill his Staph aureus. Well, if we look over here, we see that his Staph aureus is resistant to amoxicillin. Amoxicillin with clavulanic acid, it's sensitive to. Cephalexin, the one I was trying, he had intermediate sensitivity to. So maybe I was using amoxicillin on him. This is not a totally true story. Okay, so just bear with me. Gentamicin, he's sensitive to, and enrofloxacin, he's sensitive to. So again, what does each designation mean? If we say that the Staph aureus is resistant to amoxicillin, that means the dose that would achieve this, it's beyond the toxic dose. I can't use it. Cephalexin has intermediate sensitivity, and so that would mean I could use cephalexin. I could try cephalexin, but I'd have to use the maximum dose. But these three drugs, amoxicillin with clavulanic acid, gentamicin, and enrofloxacin, he's sensitive to. In other words, these doses, I can achieve these concentrations. Let me say that again. I can achieve these concentrations 4 micrograms per mil, 0.5 micrograms per mil. We can achieve that just by using the usual dose on Gordo. We don't have to use some really, really high end of the range. So which one did I choose? Well, I actually chose the Clavamox. His Staph aureus is sensitive to Clavamox, right? So even at a usual dose, it will kill his Staph aureus. And you don't know this yet, but you will over time. Clavamox is a lot cheaper than gentamicin and enrofloxacin. Clavamox is a liquid. It could go in his mouth. Gentamicin comes as an injection. I don't want the owner to have to poke her cat twice a day. And enrofloxacin is a pill, and it's no fun giving a cat a pill. So we chose a liquid, and it actually has less side effects or less potential side effects than the other two. Now, what if his Staph aureus had had a intermediate sensitivity? Well, I might have still used it, but I probably would have instead chosen enrofloxacin instead. If possible, you want to choose the drug that they're sensitive to. All right, so just to test our understanding, this comes from question number 4 in the back of chapter 10. So that little horse there has a chronic wound. He's not healing. It's not healing. You do a culture and sensitivity test. It comes back, indicates that the MIC of a drug, we'll call it drug A, the MIC of drug A needed to inhibit the bacteria is 10 micrograms per ml. And then the report also tells you that the break point for susceptible and intermediate is 4 micrograms per ml. The break point for intermediate and resistant is 16 micrograms per ml. So knowing that, should this drug be used to treat the infection, assuming the drug can be delivered sufficiently to the injection site? So what do you think? You can pause it. Is the answer yes or no? Well, the answer is yes. We could use it, but you can see that it's greater than the break point for the sensitive and for sensitive intermediate. So this would be considered intermediate sensitivity. We could use this drug, but we'd have to use it at the highest possible dose. So it would be better to find a drug that this bacteria is more sensitive to. All right, let's talk next about how are bacteria resistant? How do they do it? So there's a few different ways, and the videos do a much better job than I can describing this. But the book talks about vertical transmission and horizontal transmission, and it's really not as complicated as it sounds. Vertical transmission is how you got your traits from your parents. Bacteria are not male and female. They don't have babies, but they do divide. I guess we could call it something like mitosis. They divide and create daughter cells. And so if this bacteria develops some sort of resistance, some special power, then all of its offspring will have it. Horizontal transmission is a little different. That's like me passing you something. If I had some special weapon and I gave it to you, you're not my offspring. I'm just handing it to you. Like, I got this really cool weapon here. Take that. Um, Bacteria can share information with their neighbors, and we don't need to know these differences here. It's just, you can imagine if one E. coli has some special power and it gives it to another E. coli and another E. coli, and now they all have it, and plus they're going to replicate. So their daughter cells are going to have it. Those are the ways that bacteria share information. And what are some of these powers? Well, one power is they can pump out an antibiotic. Some antibiotics enter the bacteria. Well, now they can pump them back out. Some bacteria develop enzymes that will destroy the antibiotic. Some have a receptor that the antibiotic will dock to, but now the receptor will change shape. Imagine a receptor that's supposed to be this shape, um, you know, on a bacteria, but now it changes shape and the antibiotic can't dock anymore. And sometimes the outer layer of the bacteria changes, and that prevents antibiotic from entering the bacteria. Why does resistance happen? Well, there's a lot of different reasons, and we're responsible for some of the reasons. Certainly, using a really strong antibiotic when we could use a more wimpy antibiotic. Um, any bacteria that survive the big guns of a powerful antibiotic are probably going to have some resistance. If we use a dose that's too low, we may just kill wimpy bacteria and leave stronger bacteria behind. If we stop giving antibiotics too soon, if we use them when patients don't need them, including in livestock and fish, we are basically killing off wimpy bacteria and leaving superbugs. And the superbugs will share their information with other bacteria. And we call all of this selection pressure, killing off the weak ones and leaving the strong ones behind. And again, this is what you'll be talking about this week online. So that's resistance. We'll talk. Let's now talk about, well, how do the actual antibiotics work? Now, I'll be honest with you. I don't really care about these two concepts. They've never been very helpful to me. I looked in 3 different books to get a good explanation, and 2 of the books don't even mention it. So I don't know why this book makes such a big deal out of it. So I am not going to ask you any questions about time dependence or concentration dependence, but the VTNE might. So we're just going to mention this. Some antibiotics work because they have to be given often enough, maybe 2 or 3 times a day, so that the concentration in the blood stays above, or at that minimum inhibitory concentration. You remember you learned about steady state, right? So certain drugs are all about steady state. We have to reach a certain concentration and stay there. I think most drugs are that way. There are some that we say are concentration dependent, where we really just have to reach this particular concentration, maybe every day or every 12 hours. And then that is enough to kill But the point is, we want to follow the evidence-based dose regimens. If it says this amoxicillin has to be given 3 times a day, we have to give it 3 times a day. We can't say, well, that's kind of a pain in the butt. Just give it twice a day. We cannot do that. We have to do what the evidence tells us to do, either at a high enough dose or for a long enough period of time, or I didn't mention it, but at a appropriate frequency. Because if we don't, we are selecting for superbugs, right? There's the Mr. Antibiotic unable to punch out superbug. And another concept, it's not about how these drugs work. We'll talk about that, but we need to also remember that you'll be treating patients that make milk, or that they themselves might be eaten, like a chicken, or the chicken making an egg, or a cow, or a pig. You might be working in that, and we worry about what are called drug residues. You've learned about those already. They can end up in meat and milk. My wife, for example, is highly allergic to sulfa antibiotics. There are many people who are allergic to penicillin antibiotics. We can use penicillin in a cow, for example, but then we need to communicate to the farmer how long to withhold milk or withhold eggs or whatever, and that's called the withdrawal period. Humans, like my wife, she could have an allergic reaction if she ate an egg that had a little bit of sulfa antibiotic. People with a peanut allergy, it doesn't take very much peanut, and that child could die. It doesn't take very much of anything to trigger an allergy. And allergies are also called hypersensitivities. Also, even if it doesn't make you sick, if you eat this antibiotic, it's selecting, it's killing off some wimpy bacteria and applying this selective pressure to create superbugs. So those are some reasons why residues are a no-no. And there's a really great website that we use called FARAD. I can't remember if we looked at this before, but it stands for the Food Animal Residue Avoidance Database. And I wanted to show you a demonstration of it. So it's farad.org. Let's see. Here we go. Okay, so we're at farad.org, and you'll be checking this out in one of the quizzes, but you'll have plenty of time. So let's say we have a cow, and we want to use a particular drug on the cow. So I'm going to scroll down here, and it says WI lookup. So WDI, that stands for withdrawal interval. And it tells me all these different species here. I thought it would tell me chickens, but it doesn't tell me chickens. So I'm just going to make something up. Let's look up goats. Let's say we have a goat, actually. And what drug are we using in the goat? Oh, we're using like moxidectin, right? So you learned about moxidectin. That's one of the macrolide dewormers. So let's say I have a bunch of meat goats, and they have hookworm, or they have Haemonchus contortus, the barber pole worm. Well, I want to send these guys to slaughter, but I need to treat the barber pole worm. So I give them all moxidectin, and it tells me that tells me the dose, and it tells me that the meat withdrawal is 14 days. So after I give them moxidectin, now moxidectin is not an antibiotic, so maybe I should show you an antibiotic. But this just tells me I have to wait 14 days before I can use, before I can send them to slaughter. But let's use an antibiotic instead. So how about, you know, tetracyclines. Well, here's gentamicin. So gentamicin we're going to learn about is one of the last ones we'll learn about today. And look, if I use gentamicin on them, I have to wait 18 months before I can send them to slaughter. It's legal to use it, but I don't really want to wait 18 months. But I'm just telling you that's the withdrawal interval for that particular antibiotic. Okay, so that is Farad. And let's just look at one other thing while we're here. How do I go back? Let's try looking up a cow. Here's I don't know, a pig. Let's look up a pig. And there's penicillin. Okay, what's the withdrawal for penicillin? There's the dose, and the pig could not be sent for slaughter for 14 days at that dose. If I used a higher range, a higher dose, and then it's actually 50 days. Right? Remember, we learned about there's different ranges. You can do a medium range. This might be a high range, the high end of the dose range. Okay. All right, that's that. Let's go back to our talk. Okay. Now let's actually talk about how do these antibiotics do what they do? How do you kill this evil guy behind me? You've met Mr. Wolbachia. A totally different antibiotic will kill him, doxycycline. That's next week. Here's that term again. We use this when we discussed antiparasitics, mechanism of action, right? That's how they do what they do. And each family has the same mechanism of action. The penicillins all have the same mechanism of action. So what are some examples? Well, and I think this is a pretty good little illustration here. So bacteria are kind of like cells. They're a little weird. They've got this wall and they also have a membrane. So some antibiotics, they're going to interfere with the formation of the cell wall. Remember that little video clip I showed you of the bacteria dividing and dividing? When they're dividing, the cell wall is very active, right? The bacteria is making all these new cell walls. Well, we can interfere with the bacteria when they're dividing like that, and they'll just burst like a popped balloon. We can some different drugs will interfere with the actual membrane, and that'll make it easier for other drugs to enter. I don't think that's very common. This third one is very common. Some antibiotics, they're going to interfere with something inside the bacteria. They're going to interfere with just some basic life process, making proteins, the functions of enzymes, right? Bacteria they have all the same needs or some of the same needs that you do. They need to make proteins, they need DNA, they need RNA, they have enzymes. So if we can mess with those life processes, then we can kill the bacteria. And this gives you some examples: inhibiting protein synthesis, inhibiting nucleic acid synthesis so they can't make DNA, blocking the production of folic acid, so things like that. So let's talk about the first of our three groups. The first are the penicillins. And they all end with -cillin. They all do. There is no exception. And they're sometimes called beta-lactam antibiotics because if you looked at all of these different penicillins, they all have this little ring. It really looks more like a square, but they call it a ring. So it's just called the beta-lactam ring, and they all have it. Penicillins are usually going to be more effective against gram positives and not so effective against gram negatives. But you'll see. How do they work? Well, they interfere with the cell wall formation. And so when the bacteria are rapidly dividing, if penicillin is there, it's going to make this happen. They're trying to divide, and they can't because penicillin messes with the cell wall, and then water comes in, and the little bacteria burst. So this is only effective when they're dividing, and you may find that when there's an abscess, there's a lot of bacteria in an abscess, but they're not dividing. They're just kind of floating in the soup of pus. So this is part of why when a cat comes in with a big abscess, we can't just send them home with antibiotics because the drug won't reach all of the bacteria. We need to make a little hole and flush it and squeeze it all out, and then the bacteria will start to divide, and then the penicillin will work. Penicillins are hydrophilic, which is just a way of saying that they cannot reach the eye, the brain, or the prostate gland. You can just think of all three of those organs as having like this fatty wall around them. And penicillins cannot cross it because they're hydrophilic. And one nice thing about penicillins is you learned how in the kidneys, they reabsorb a lot of good stuff. Well, the kidneys will also excrete or secrete a lot of bad stuff. That's why employers have you do a urine test for drugs, because a lot of drugs are excreted in the urine. Well, the same goes for most antibiotics. They will end up in the urine. Well, that's great if you have a kidney or urinary bladder infection, right? Now we've got these really way higher levels than we can get anywhere else. We can get in the kidneys and the urine, and it's still active. And so we can sometimes use antibiotics to treat bladder infections. That if that infection were on the skin, we wouldn't do it because the antibiotic could not reach such crazy high levels. But in the urine, it can reach crazy high levels. So that's kind of cool. There are sort of 4 categories of penicillins. One are called the natural penicillins, like Pen-G. That's what I have here. These will often have something like procaine added to it. Sorry, that doesn't really show up. Oh well, sorry. Procaine just makes it last a little longer. There are some that are a little more broad spectrum, like amoxicillin and ampicillin. These will actually work a little bit better against gram negatives, but these guys, they will only work against gram positives. And I wanted to say something that there are some bacteria that are resistant to these first 2 groups. So there are some bacteria that just won't work. Some I should say some gram positive bacteria, because they have the ability to destroy that ring. Right here, this little guy is a kind of a staphylococcus called Staph aureus. And remember, I said aureus means gold. Right, so here's Staph aureus, and these guys make an enzyme called beta-lactamase, and that enzyme destroys the ring, and so he just laughs at penicillin. Well, they made some antibiotics that that can resist the beta lactamase, and beta lactamase is the same as penicillinase, and so these are called penicillinase resistant penicillins, right? They would be very effective against Staph aureus, and they have names like cloxacillin and dicloxacillin and oxacillin and methicillin. Now, the book says those are the most commonly used ones in veterinary medicine. Um, um, I see a lot of cool things in general practice, a lot of emergencies and complicated cases. I have never worked at a clinic, and I've worked at probably 5 different clinics um, doing relief work and full-time work. I have never seen any of those antibiotics. The same goes for these ones called extended spectrum penicillins, ticarcillin, piperacillin. I've never seen these. Now, I think if you went to work at an emergency clinic, or if you worked for a specialty clinic, a referral clinic, you would probably see these. These are very powerful penicillins. I'm not going to ask you, I might ask you a couple of questions and do the homework, but I'm not going to ask a bunch of questions about these. I think it's more important to learn other things we're talking about. Okay, so there's some general characteristics that we should know about the penicillins. One is that if a bacteria develops resistance to one type, they can often develop resistance to all types. Now that's a little bit of an overstatement. It's sort of, but it's like we said, Staph aureus can destroy this antibiotic, this penicillin. Well, that means it can destroy this one and several others. So we just call that cross resistance. It's not like, well, he's resistant to amoxicillin, let's give him ampicillin. It won't work. The other really kind of cool thing, this is probably my favorite thing I'm going to tell you today, are there's one kind of penicillin called the potentiated penicillin. And that would be when we add something like clavulanic acid or sulbactam. And we're just going to talk about clavulanic acid. And what happens when you add clavulanic acid to something like amoxicillin? Okay, so remember we said here's all of the penicillins have this ring, and we learned that certain bacteria like Staph aureus can attack that ring. Well, we can add clavulanic acid, also called clavulanate. You'll notice it has a beta-lactam ring also. So if we add this is not an antibiotic. It's just something that kind of looks like penicillin. And what will happen are the Staph aureus or whatever beta-lactamase bacteria, they'll attack this ring by mistake, and it sort of tricks the bacteria, and then that leaves this penicillin free to kill Staph aureus. I love it. I love tricking bacteria. And then the last thing we'll say about the penicillins is there is one particular resistant bacteria known as methicillin-resistant Staphylococcus aureus. It's for short, it's MRSA, and this Staph aureus has developed resistance to all penicillins. Clavamox, he's not fooled by the clavulanic acid doesn't matter. The reason this particular bacteria is resistant to every penicillin is because the cell wall that is the target, that the part of it that it's an enzyme, the part that the penicillin wants to dock with has a totally different shape. I put these little squares here to be penicillins, and I put these little shapes here. You see, they're not square, so the penicillin can't dock to Staph aureus, and if it can't dock, then it can't disrupt the formation of the cell wall. And so MRSA are horrible. We have to use a completely different type of antibiotic to kill them. And I'm sorry, there is one more thing about penicillins are the adverse effects. Now, in most patients, amoxicillin, Clavamox, there's very few side effects. They might cause a little upset stomach, maybe a little vomiting, a little diarrhea, but generally these are very safe. Some individuals are allergic to them, and that includes horses, dogs, and cats and cows. So they can break out in hives. You might know them. The other term would be urticaria. They can have anaphylaxis. In 25 years, I've never seen an animal have an allergic reaction to penicillin, but it can happen. The other concern is in certain species, we either cannot use or we try to avoid using penicillins because the problem with antibiotics is they can kill the good bacteria as well. One of the videos said you have more good bacteria in your intestines than cells in your body. So you have a whole population of good bacteria. The antibiotics, they can't always tell the difference, right? They're just going to kill whoever is susceptible. And so something about guinea pigs and rabbits and hamsters, perhaps they have a lot of gram-positive bacteria, but the penicillin can wipe out so many of their good bacteria, the good gut microflora, that whatever's left behind takes over and they get really severe diarrhea and they can die. So like in guinea pigs, we would just never, you could kill a guinea pig if you gave it penicillin. Whereas it could, it could help a goat or a dog. And I don't know if this is true, but I thought it made the point well that back in 1929, I don't know if they used mice or what, but they're saying if they'd used guinea pigs to test penicillin, they might have thought, oh, this stuff's poison, don't use it. Now this I put on here, this is not for testing, but I thought I know what it's like when I first learned these antibiotics. You're like, they all sound the same, and what's the difference? And so, but as time goes on, you really start to develop a fondness for them. Like, oh, thank you, ampicillin, for saving this patient. So I just thought I'd give you some examples. So let's say you had a patient who licked their incision. It's infected, but it's not nasty. I'm going to use the word nasty a lot. So it's infected, it's a little gooey, but it's probably just, strep and staph bacteria, either already on the skin or in the mouth that have just caused an infection. So I'd probably use something like amoxicillin, right? It's very effective against most simple gram positives, and there's nothing nasty there. And amoxicillin, in general, it comes in a pill form. An animal that has a little puncture wound that's pretty fresh, well, I don't want them to get tetanus. Amoxicillin is also very effective against tetanus. So I might use, kind of simple, fresh things, fresh wounds that aren't too bad. Now, if a patient were undergoing orthopedic surgery, now we have we have their joint exposed to room air. I might use something similar to amoxicillin. I don't think amoxicillin comes as an injectable, but I can use ampicillin. Ampicillin is an injectable. I could give it IV every hour or so. just in case any bacteria tries to get into that joint, into that bone, the ampicillin will help kill it. And then finally, if a cat came in with kind of a nasty cat bite abscess, right? It's not like this wound. Right now, it's, a cat bit this cat. So who knows what's in there? Probably some pretty nasty bacteria. And so for that cat, I might use something like amoxicillin clavulanic acid. If I'm concerned that there's, and by nasty, maybe I mean that it could be resistant to amoxicillin. And there may be a big variety. I don't even know what's in there. I just want something that will cover, most gram positives. Okay, any questions on that? The next drug, you're going to like this. They are basically the same as the penicillins. The beta-lactam ring, they interfere with the cell wall. They can't penetrate the brain or the prostate. I don't know if they reach high concentrations in the urine, but that's not important. And they actually appear, the one difference is they do appear to be a little bit safer for rabbits and hamsters. But basically, these are the same as penicillins. They're very, very similar. The non-proprietary name will always begin with the letter C. Cefazolin, cephalexin, ceftiofur, cefpodoxime, cefovecin. They will all start with a C. You might know, I put Keflex there just because some people have taken Keflex. That was a cephalosporin. This is a little pill that's very, very similar to cephalexin. Cefpodoxime is once a day. Cephalexin would be twice a day. I actually have some cefpodoxime right here. I know you probably can't see that, but it's just a nice little pill, right? Oh, who cares? Here's a little funny story. I was talking to when I was in veterinary school, we had a conversation. If the zombie apocalypse came, and you only had one antibiotic you could have forever, which one would you choose? We all like the cephalosporins. They're great at gram positives. There are some that are actually pretty good at gram negatives, So anyway, the Convenia is a cephalosporin that is an injection that lasts, I want to say 7 to 10 days. So it's very convenient for the owner. We just give the shot at the clinic. They're done. Cefazolin is kind of like ampicillin, something we might use during a surgery. If I had a patient with parvo, I might keep them on cefazolin or ampicillin while they're in the hospital because they are so immunocompromised. And the cephalosporins are divided by different generations. We only use the first generation and the third generation cephalosporins. The oldest one, that's the first generation. That would be like cephalexin, right? Good old Keflex here. Really only good against gram-positive bacteria, but pretty good against even like Staph aureus. It can work pretty well. The third generations are things like the Convenia, cefazolin. They're still very effective against gram positives, but they are a little more effective against gram negatives. So if a patient had E. coli or salmonella, we might be able to use these. And I basically told you when we would use them. So these are animals with skin infections. These would be great for giving them an injection of Convenia or any cephalexin. If I used amoxicillin on these, I might just be worried that it won't be enough. Clavamox would be fine. Anyway, okay, and now we're almost done. Our final antibiotic for this week are the aminoglycosides. And if you've ever used Neosporin, you used an aminoglycoside. If you've ever used an ear ointment on a dog or cat, take a look at the bottle. It might contain an aminoglycoside. I'll show you an example. But the neomycin is the aminoglycoside. These are really powerful. They're awesome, but they're kind of old, and you'll see why that matters. You'll learn next week about a different group of drugs um, that have kind of replaced these. But for 20, 30 years, this was the big gun. If we needed to kill E. coli or salmonella or some really nasty gram-negative bacteria, we used aminoglycosides. So gentamicin, amikacin, those are probably the two most common in veterinary medicine. They do not come in a pill form. They just don't get absorbed well if they're eaten. So they can be given as an injection if you need to get it into the body. We use them to treat gram-negative infections of the eye or a gram-negative infection of the ear, like Pseudomonas. If a patient had Pseudomonas in their eye or in their ear, this is probably what we would use. It's absorbed pretty quickly. A problem, though, is that you'll see other drugs that end in mycin, MYCIN, that they are not aminoglycosides. So just keep that in mind. This one works by going inside the bacteria. It's going to interfere with a life process. It also has something called a post-antibiotic effect. So even after it's reached its peak concentration, it'll keep killing. Kind of cool. Cannot penetrate the brain or the eye, or I assume the prostate. So we can't use it for prostatitis or meningitis or anything like that. You can put it, that's giving it as an injection. If you have an eye infection, you can put drops on the eye. That's different. When I say it cannot penetrate the brain or eye, I mean if you gave an animal an injection, it would never reach, it would get into their bloodstream, but it would never reach the eye. I'm always looking off at that side of the screen, so I'm sorry, I'm trying to look at the camera. The aminoglycosides, they're actively transported into the ear and into the eye. If you put drops on the eye or drops, oh, I'm sorry. It's not transported into the eye. It's if you give an animal an injection, it will be it'll be distributed throughout the body, and it'll achieve high levels in the kidneys and the inner ear. The inner ear is where your cochlea is. That's where you actually, that's where the not the optic nerve, but the nerve that transmits sound information to your brain is all found in the inner ear. So this concept of active transportation may help you remember something. Active transport requires ATP, it requires oxygen. And what it means is it can pump aminoglycosides against their concentration gradient. So even if there's a lot of aminoglycoside in the ear, more can get pumped in. Okay. And actually what I should say is it gets actively transported everywhere. And that's important because active transport requires oxygen. That means these drugs only work in parts of the body with oxygen. Some parts of your body don't have a lot of oxygen. Your colon, there's not a lot of oxygen in your colon. There's not a lot of oxygen in a deep puncture wound or in an abscess. So these wouldn't work that well If you flushed the abscess, they might work better. But if you had some sort of a infection in the colon, this would never reach it. It would never work. The other thing about the fact that it can achieve high levels, in particular in the ear and in the kidneys, has to do with some of the adverse effects. So this drug is potentially capable of harming the kidney. And the word we use is nephrotoxic. Nephro is a term that means, well, it refers to the nephron, really. The nephron, the loop of Henle is part of the nephron. We learned about the nephron a few weeks ago when we learned about cardiac drugs. But that's your kidney is made up of just hundreds of thousands of nephrons. So these drugs are potentially dangerous and harmful. We can use them, but we got to make sure the patient is not dehydrated. Maybe we'll use IV fluids to help flush any aminoglycosides from them. And if we were using it in a young animal, we might make sure we use the lowest possible dose. This ototoxic is truly important. It's all important, but the most common time that we use these drugs are in ear ointments for patients with an ear infection. And you see right here, this is an ear ointment called Mometamax, and it contains gentamicin. And we put that in a patient's ear. If that patient had a ruptured eardrum, we could cause permanent deafness. And in a dog, deafness is not good, right? The dog cannot hear its owner. If the dog's running into traffic and a car is coming, the dog can't hear the car. So we cannot use these drugs unless we can see the eardrum. Or what I should say is we really should see the eardrum, and it should be a perfectly intact eardrum. Some dogs, their ears are so painful that we can't look, and then we have to tell owners, well, we can sedate him and look at the eardrum, or we can take our chances and use this. But if the eardrum is ruptured, this could cause deafness. Now, luckily, the eardrum is almost never ruptured, but we need for the client to make that decision. They need to know there's a risk. It might be very small, but there's a risk that this could cause deafness if the eardrum is ruptured. Okay, that was it for part 1 of antibiotics. To review what you've learned, I'm going to create a little Quizlet for you. So you can go and check that out. I'll probably have it up tomorrow. And then that is all. If you have any questions, email me. Start to work on the self-assessment questions in the back of the book. And you can also call me, of course. So I hope you're all doing well. And I will see you online. And I'll see you next week. All right, take care.